So, have you ever actually stopped to wonder how on earth scientists study the sheer chaos of millions of microscopic cells interacting? I mean, think about a growing tumor for a second. You've got these incredibly complex overlapping dynamics happening all at the exact same time. Cells are moving, they're dividing, releasing chemicals, dying off. Trying to observe all of that action at scale under a standard microscope, yeah, that's practically impossible. Well, today in this explainer, we're going to dive into a truly revolutionary piece of software called PhysiCell. It's an open-source, physics-based cell simulator, and essentially, it allows researchers to build this massive, fully 3D virtual laboratory right there on their own computers. Here is our quick roadmap for today. We're going to look at the multicellular challenge, the stage and players, and then we'll peek inside the PhysiCell engine. Finally, we'll cover solving computational bottlenecks and why PhysiCell matters. So we're going from pure biology straight into computer science. All right, part 1, the multicellular challenge, modeling complex tissues and tumors. To really get why PhysiCell is such a big deal, we have to recognize the massive hurdles of the older simulators. You see, platforms like CompuCell 3D or Morpheus, they're super user-friendly, but they hit a brick wall when trying to scale past, say, 100,000 cells. Then you have others like BioCillion or Tysim, which can handle the massive scale, but they're locked behind strict closed-source licenses. And then tools like Chased, they have these incredibly complex code-based dependencies that honestly make them a total nightmare for new developers to adopt. PhysiCell was engineered specifically to smash right through all of these bottlenecks. The developers actually tackled this challenge using a pretty brilliant theatrical metaphor. They realized that to build the ultimate virtual laboratory, you have to accurately simulate two very distinct things. First, the biochemical microenvironment, which they called the stage, and second, the individual cells themselves, which are the players acting upon that stage. Which brings us right to part 2, the stage and players, focusing on bio-VVM and agent-based cells. First up, let's build that stage. PhysiCell is built on top of a lightning-fast biotransport solver called BioFVM. Imagine this complex 3D environment where chemical signals are constantly shifting around. You've got oxygen diffusing through tissue, nutrients being gobbled up by hungry cells, and maybe targeted cancer drugs decaying over time. BioFVM is the engine that calculates all of that dynamic background chemistry. Now, right onto that stage, we drop the players. These are our individual cell agents. And what makes PhysiCell so incredibly powerful is what it handles right out of the box. Instead of forcing you to code basic biology from absolute scratch, researchers immediately get cells that natively just know how to progress through their life cycles, how to change volume, and how to move around. It even includes the hardcore math for two very distinct forms of cell death, apoptosis and necrosis. And the way the software models these two types of cell death, the mathematical precision is just wild. If a cell undergoes apoptosis, which is programmed cell death, the code dictates that it naturally eliminates water and dramatically shrinks. But if it experiences necrosis, which is an unprogrammed, messy death, usually caused by oxygen starvation, the math triggers oncosis. The cell essentially swells up until it eventually bursts. It's a truly incredible level of digital biological accuracy. All right, let's move on to section 3, inside the PhysiCell engine, where we look at the mechanics of the simulation. So you might be wondering, how exactly does this system manage all of these moving parts without just instantly crashing your computer? Well, the secret sauce here is decoupled time steps. Think about it. In actual biology, chemical diffusion happens in an absolute flash. Cell movement, that's a bit slower. And cell division, that is a very, very long process. So PhysiCell actually separates its calculations to perfectly match reality. Rapid updates for chemistry, medium updates for movement, and super infrequent updates for cell division and death. To put some numbers to that, here is how it breaks down mathematically. Biotransport is calculated every 0.01 minutes. Cell mechanics, those update every 0.1 minutes. But those complex cell lifecycle processes, they are only calculated every 6 minutes. By flat out refusing to calculate every single biological process at every single microsecond, PhysiCell frees up just a massive amount of computing power. Which leads us nicely into section 4, solving computational bottlenecks and scaling up with OpenMP. To actually simulate millions of cells smoothly, the software relies on a really elegant 5-step program loop. Step 1, save the simulation state. Step 2, run that lightning-fast chemical microenvironment. Step 3, run the much slower cell lifecycle processes. Step 4, calculate the mechanical forces, where cells are physically pushing and pulling on one another. And finally, step 5, update the simulation time. and loop right back around. Now, step 4 is usually where most other simulators just completely freeze up. Look, if you have a million cells and every single cell has to mathematically check its distance against every other cell in the entire simulation just to see if they're touching, the computational cost scales quadratically. The math just becomes utterly impossible. But PhysiCell uses a very clever data structure. A cell only bothers checking the immediate space right around it, its bounded local neighborhood. And that one tweak, it mathematically drops the computational cost from quadratic all the way down to linear. It's brilliant. And the real-world result of that optimization is genuinely staggering. 500,000. That is the number of fully interacting, highly dynamic 3D cells that you can smoothly simulate on a standard, everyday quad-core desktop workstation, literally right on your desk. Take that exact same software and deploy it on a single high-performance compute node. It scales linearly to over 1 million cells. That's 1 million individual agents, living, dividing, dying, and secreting chemicals. And it's all being processed simultaneously without the computer even breaking a sweat. And that brings us to our final part, part 5, why PhysiCell matters and how it's democratizing multicellular research. Unlike those older proprietary models that locked all their code away or required literal millions of dollars in supercomputers, PhysiCell is completely open source. It's cross-platform, it's written in standard C++, and it has minimal external dependencies. And what that really means is that anyone, from a massive, well-funded university lab down to a small undergraduate classroom, can just download it and start experimenting immediately. And the real-world applications here are absolutely vast. As we speak, researchers are using PhysiCell to simulate cancer immunology. They are literally watching digital immune cells physically hunt down digital tumor cells on screen. They're engineering synthetic multicellular systems for highly targeted drug delivery. But maybe most importantly, it allows for super high throughput hypothesis testing. A scientist can rapidly simulate thousands of different scenarios digitally in a single afternoon, way before spending months and millions of dollars testing them in a physical wet lab. So we now have this incredibly powerful, completely open source virtual lab, one that is fully capable of simulating millions of complex, interacting cells right on a standard computer. The tools to decode the chaotic dynamics of human tissue are finally in everyone's hands. The only real question left is, what biological mystery are we going to solve next?